Bio 30, 19.2 today, causes of gene pool change, a really quick lesson of just basically the 5 agents of change, the things that can limit or expand variation in a population. We know that diversity is how much variation there is in that population, and these changes in the gene pool can happen because of these 5 things. One of them, and you guys will recognize a lot of these, one of them is mutations, which can happen when DNA is altered. Many times it's a negative change, sometimes it's a positive change. So it depends on the environment that organism is living in, whether that mutation will give them an advantage for survival or not. And if there's something that is inherited by future generations because it was positive, it will then diversify the gene pool and be found in the gene pool. So in this rat, the Norway rat, the advantage that it had, these examples I put in here, but they're all in your textbook, is one mutation was that it was resistant to rat poison. And of course that kept it alive. So that was passed on to future generations. So mutation is our first one. Second one is gene flow. And gene flow is the movement of those alleles from one population to another because of organisms migrating. So they will go and mate with other populations. So just think about maybe coconuts flowing from one island to another island. So think of that as gene flow. And as basically it says here, cultural and geographic barriers are not so stringent anymore. Individuals from some cultures will have offspring with individuals from other cultures. And so that would be an example of gene flow. In the wild, animals tend to naturally mate with nearby populations instead of within their population. And so that will bring new alleles into their gene pool. Third one, non-random mating, is means in which typically happens in nature is that that it's not random that they mate with just any individual. It's typically non-random because there will be a preferred phenotype. So depends on the organism, but like the sage grouse prefers the mate to have really bright and tall long tail feathers and a big air sac in the front. So that's an example. There are preferred phenotypes that the individuals will have, and they are basically valuing specific features or traits in their opposite sex. And an example with that too is that in terms of human beings, there is a large majority that mate within their ethnic group. Inbreeding can be negative because sometimes when individuals within the same group mate, there's more likely to be the recessive, maybe even deadly or disease-causing alleles that may end up together and be expressed. So we looked at how that happened with like hemophilia in royal families that tended to mate within their family. Another thing that could happen is that some alleles could be eliminated if they mate that way. So in goats, inbreeding has caused expression of an underbite, and that's kind of common with them if that happens. And of course, close relatives with kids, they tend to have that recessive trait more likely expressed. Okay, fourth agent of change is genetic drift. Now, gene pool was something that happened when genes flowed from one population to another as they mixed. Genetic drift is a chance event, and there are 2 examples of a chance event that could happen. One of them is the founder effect. And the founder effect is, think of a group who finds an island and decides to settle there, and basically they only have this small number of individuals to mate with. The founder effect says the founders will have this limited gene pool, and often happens in small groups, islands, or colonies. And what happens there is then if there is some recessive condition like the Amish who have a more prevalence of Ellis Van Creveld syndrome, this is what is going to cause that to be exposed. And Ellis Van Creveld is a dwarfism with polydactyly, many fingers and toes, and can be associated with heart defects as well. Second kind of genetic drift is the bottleneck effect, and that is when the gene pool is drastically reduced because of something that, again, a chance event that happened that causes them to reduce in numbers. It could be some disease, it could be whatever agent of extinction, could be overhunting. But when a whole bunch of them are lost, the gene pool that had many genes in it becomes a small gene pool, and that's another example of genetic drift. So that's founder effect, where there's a small group that found an island, for example, and genetic and bottleneck effect, where a bunch of them have been eliminated. The cheetahs are being affected by the bottleneck effect. And of course, bottleneck means there was a lot, and it's like an upside down bottle where there's much less after. And the cheetahs, because of overhunting, have a much lower genetic variation, and that's causing infertility issues and a big concern with cheetah populations. So with bottleneck, as we only have a few left, they may, in this example, which used to be both alleles, lots of yellow and blue, if there's a small amount left and there's only a couple yellows, that means the future generation will not have many yellows, if that was a certain allele. Okay, last kind of agent of change is familiar to you from Bio 20, that's natural selection. And basically, if there are favorable variations that allow them to adapt to their environment, give them some selective advantage, then those are the organisms that will live to reproduce and have babies and give those traits to their offspring. So sexual selection is when a certain phenotype attracts a mate. And one interesting thing is that some lethal alleles end up staying in the gene pool if that individual has some survival advantage because it carries that allele. So these three are interesting examples here that the individuals with cystic fibrosis have a resistance to cholera. Individuals with Tay-Sachs, which is a nerve damage, have a resistance to tuberculosis. And individuals with PKU, which is a nervous system seizure disorder, will have a resistance to prenatal fungal toxin. So these are just interesting examples where when people are heterozygous for these conditions, they have an advantage. They're carriers, but they have an advantage. So that ends up when those conditions or those diseases are prevalent, they survive. And so those conditions keep getting passed on through the future generations. Anyways, natural selection, guys, is when the genes continue through the population because they have some advantage, they help that individual survive. So this is just a picture of the 5 agents of change that we have discussed. Mutation, that may give an advantage. Gene flow, which is reproducing with nearby populations. Non-random mating, which is when certain phenotypes are more likely to be attracted to the opposite sex. Genetic drift, which was the founder, which is small group on an island, or the bottleneck effect, where there were only a few left because a bunch were wiped out. Those are two examples of genetic drift. And natural selection, where the ones best suited to the environment survive. So the green ones here, camouflage and survive. So those are the 5 agents of change. That wraps up our topic. There's 2 last slides just talking about threats to declining diversity, things like habitat loss, fragmenting the wilderness, causing it to be in pieces because of roads going through it, for example. Over-harvesting could be overhunting, for example, or loss of traditional crop varieties, which is why we have seed banks, for example, that are trying to maintain that. And to protect this, we have natural preserves, parks, treaties, protection of endangered species, protecting their habitat, seed banks, gene banks, and breeding programs to try to preserve all the species that we know of that we have on Earth today. Okay, thanks for joining me